Hey guys, how you all are doing? I hope you all are doing really fine. So finally we got to start with staining of organisms, and while listening to staining of organisms, there are two questions that comes to our mind. What is staining and why do we need it? So the procedure of making translucent organisms colored or stained is called staining, and we need it because translucent organisms cannot be studied under microscopes properly. So to make the translucent organisms visible, we need to stain them. Moving ahead. So what all we gonna study in this part? In this staining series, so the introduction, the types of stain, solutions and reagents used during staining procedure and staining techniques. Now, staining techniques like acid fast staining, gram staining and all, staining procedure will be different for light microscope and for electron microscope. There's some solutions and reagents used during stain. So like the if you have heard about mordant and all, like, so these are the kind of terms that we use to name the solutions and reagents that have particular functions. And there's some types of stain to stain different types of organisms. So let's go ahead with the introduction of the staining part. So, as we have discussed in the previous section, why to stain is of course because transparent organisms are not visible, and though there are some slight differences in the refractive index of cytoplasm and the materials or the structures around it, though those differences are not sufficient to make the organism properly visible under the microscope. What happens after staining? They become visible. The shape, size, and arrangements are become visible. Internal and external structures can be studied, and taxonomic identification is also the big thing that staining help us to do. How? Let's see. We have a picture of stained bacteria over here. We can see that these are rod-shaped bacteria. Now, when these are rod-shaped, we can say that these belongs to the class Bacilli, because Bacilli class includes all rod-shaped bacteria. So that's how staining helps us to do the taxonomic identification. Now the two things which are used alternatively, dye and staining, they both are used for coloring, but dye is used for coloring of non-living things like your hair, your t-shirt or maybe some other clothes, and staining is used for coloring of living tissues like bacteria or maybe other organisms. I hope that's clear. Though dye and staining are not that hard and fast rule, but that's a correct term to use. Now, if you think that stain comprises of just one part, you're wrong. Stain comprises of two things. Stain is doing mainly two things if you say. First, it is binding to the organism, like say if this is a stain, it is binding to the organism here, making bond with it, and also imparting color to it. So we need to have two main parts of the stain. First is your chromophore that imparts stain to the color, that imparts color to the organism. And if we are using chromophore to stain the organism, it will not be able to impart color because it has no ability to bind with structures. Okay, let's see, we have a trinitrobenzene chromophore here. Basically it's a chromogen. Why? Because benzene is connected with a chromophore, so that becomes chromogen. So benzene with any chromophoric structure becomes chromogen. So we have a chromogen over here. So if we use trinitrobenzene to color bacteria, it will not be able to color the bacteria because it has no ability to bind with the structures. And the other one is Auxochrome. It has a property of electrolytic dissociation, or we can say it has the property to bind with the structures. Like say we have OH in here. Okay, in picric acid, when we add OH to trinitrobenzene, it becomes picric acid. So picric acid is a stain which imparts color to the organism because of its chromophoric material, chromophoric part, and bind with the organism because of the auxochromic part. I hope that's clear. So basically a stain comprises of two parts. One gives the color to the organism and the other binds with the organism. So the both of them are insufficient, but a stain completes that insufficiency and forms a proper material to stain any organisms. So that's it for this video, guys. Thank you so much for listening me out. In the next section, we're of course going to talk about types of stain. So till then, stay tuned, hit like and share button. It'll be highly appreciated. So thank you so much. Take care. Bye bye.